In these hills lies a record of life on Earth dating back 19 million years, to a time long before the first human eye gazed over this valley. It is a record written in fossilized bones and the rock that surrounds them. Under a sky hazy with volcanic ash, hundreds of animals perished here. The fossils tell their story, if we can only learn to read it. In the 1880s, James Cook, a local rancher, discovered something near the two conical hills we call the Agate fossil beds. Cook was not a paleontologist, but he was a curious man interested in the scientific explorations of his day. He found something remarkable, what he imagined to be the petrified bones of horses. Years later, in 1892, Cook invited Dr. Irwin Barbour of the University of Nebraska to the Agate Springs Ranch to investigate his discovery. Barbour was the first of many scientists who had come to the Agate Springs area. Dr. Barbour became fascinated by the mysterious devil's corkscrews, or demonelix, spiral formations scattered throughout the Niobrara Valley. He thought they were the fossilized roots of giant prehistoric plants. But paleontology is like a detective story in which new clues can shatter existing theories. The demonelix were only one of the mysteries at Agate, and when Dr. Barbour moved on before exploring further, he narrowly missed the find of a lifetime. More than a decade later, Olaf Peterson of Pittsburgh's Carnegie Museum expanded on Barbour's work. Within some of the devil's corkscrews, he found the fossilized bones of Paleocastor, a mammal who ate plants and dug burrows, much like the modern prairie dog. The spirals were not roots at all, Peterson realized, but the sandstone-filled dens of Paleocastor. At Cook's request, Peterson turned his attention to the site of Cook's original find and was amazed by what he uncovered. Peterson recognized a bone bed, a 2-foot-thick layer of rock densely packed with the mineralized skeletons of hundreds of prehistoric mammals. Confronted by the bones of strange, unknown creatures, Peterson and his colleagues from Yale, Amherst, the American Museum of Natural History, and the University of Nebraska began the painstaking, sometimes startling process of reconstructing the ancient past. What were these ancient animals? How did they live? What kind of environment sustained them? And how did they die here? The scientists knew this much. The fossils were concentrated east of Cook's Ranch in the two rocky mounds that came to be called Carnegie Hill and University Hill, after the scientific teams that first excavated them. While bones of more than 30 species of prehistoric mammals have been found in the same geological formation as the Agate bone bed, most of the Agate fossils belong to only 3 mammals, species that had never before been found in such large numbers or in such good condition. The most abundant mammal found was Monoceros, which resembled our modern rhino. Its teeth tell us it ate plant material, possibly grass, and it may have been a browser like the modern rhinoceros. The male might have defended itself from predators with the 2 horns at the end of its snout. The large number of these beasts found here together suggest they may have moved in herds. Second in abundance was another plant eater, the horse-headed Calicotherium meropus. It may have used its long front legs and clawed hooves to reach leaves and trees, though no fossilized trees have yet been found here. Whatever caused these herbivores to die off in such large numbers also claimed many scavengers. Ready to pick over the bones of its fallen neighbors was the Entelodont Dinohyus, a 6-foot-tall, pig-like beast whose teeth show an appetite for both plants and carcasses. Some bones in the bone bed show the marks of this omnivore's bite. Lying in wait for the youngest and weakest of the plant eaters was the bear dog Diphenodon, a wolf-like mammal bearing the sharp teeth of a meat-eating predator. It was not until the 1980s that a team from the University of Nebraska discovered that these and other carnivores lived in underground dens. Animal bones found in the dens revealed that these hunters preyed upon young rhinos, camels, and sheep-like oreodonts. The early paleontologists placed these animals in the Miocene epoch, which spans the time from 5 million to 24 million years ago. Modern fission track dating tells us more accurately that the mammals in the bone bed lived 19 million years ago. The Demonelix burrows are older, dating back 21 to 22 million years. These animals are more recent than the dinosaurs, who roamed the earth 65 to 225 million years ago, but much more ancient than human beings, whose ancestors, scientists believe, first appeared about 4 million years ago. Thanks to Olaf Petersen's perseverance in studying, mapping, and excavating the hills, Agate emerged as one of the most scientifically interesting sites in North America. Agate fossils appear in museum collections throughout the world. Paleontology was still a young science when these bones were unearthed. So, while they represented impressive trophies and provided the first look at complete skeletons of these mammals, still the animals lacked context. Scientists understood little about their environment or how they came to be in the bone bed. At the beginning of this century, many questions remained unanswered. But since then, the science of paleontology has gone through an evolution of its own.
Today we know that if we pay close attention to the rocks, we study them both with microscopes and we study them in the field with the naked eye, we can reconstruct the environment to an extent much greater than we'd ever thought of in the past, certainly much greater than paleontologists believed in the early 20th century, at the time the quarries were first discovered.
The rocks reveal tiny fossilized grass roots. No actual fossilized grass has been found, but scientists surmise that the grass was tall because of the presence of small mammals like the tiny sheep-like oreodont, which would have needed tall grass to hide from predators.
Now, when we reconstruct the events that produced the bone bed, we work backwards in a sense. We start with the bones and the sediments, we make observations and reconstruct the puzzle one piece at a time.
Modern scientists find clues not only in the ancient rocks, but in the modern techniques of statistical analysis. By studying groups of fossils in this way, we can learn about the relative sizes, ages, and living habits of entire communities rather than just individual animals.
Here at Agate, there are whole populations, hundreds of mammals preserved intact that must have been part of a common gene pool, if not part of a single herd that actually lived together. So here was a chance to reconstruct the nature of a fossil population 19 to 20 million years old.
Bones of Meropus and Monoceros calves have been found in the bone bed together with adults. Something devastated the young and the old at once. But what? To answer these questions, we turn to another continent, 7,000 miles away.
The environment that we try to reconstruct here may not have existed exactly in the same form anywhere in the world. But the closest modern analog that we can probably recognize is an environment similar to the Serengeti Plain in Tanzania and into Kenya in East Africa.
The Serengeti is a flat, grassy savanna. Wide, shallow streams meander through the tall grass. Herds of mammals roam, hunt, and graze. The climate is seasonal, with dry spells followed by periods of intense rainfall. In the Serengeti, we find not only the right type of environment, but we can actually see entire herds of animals dying near water holes during droughts.
There's still water in rivers and in water holes, often a very limited amount, but it's still there. But the animals die of malnutrition because they eat all the vegetation in the proximity of the water hole and gradually work out from that water hole or that river farther and farther until they use up all the vegetation that's available to them. Finally, the distance between the water hole and food becomes so great that they can no longer make that distance effectively with the strength that they have left. And so they return to the water holes in many cases, into the rivers, and lie down in that soft mud, which is cool and still has some moisture, and finally expire.
This more developed picture is by no means the final one. For over 100 years, paleontologists have built on the work of those who came before them. They continue to put together a picture of the past meticulously, one piece at a time.
The history of evolution is a painting, a fabric that's reconstructed of many strands, each one of these mammal species, plant species, that we can study out here to put the whole picture of evolution together. Agate is just a small piece of the evidence in the picture that tells us that we're part of this planet and we're part of these changing resources. And you do have a lot of insight in that sense as to what the human condition is and what's happening to man at the present time.
More than 100 years ago, James Cook found something truly extraordinary jutting out of these hills. Today, the more we learn about the agate fossil beds, the ancient mammals entombed there, and the time and history they preserve for us, the more clearly we recognize the enormous value of this place and the need to preserve it. For just as we are inspired by the first scientists who came to Agate Springs, tomorrow's scientists will build upon our curiosity and sense of wonder.
What you hope is that the next generation is going to come along and they're going to find new evidence to fill out that picture where you stopped.
In learning to read the fossil record, we are striving to understand the past and future of the Earth and of life itself.